Welcome to the chapter Nutrition in Plants.
This slide presents the overview of the chapter. Learning objectives. By the end of this chapter, you will be able to
define nutrition and modes of nutrition. Describe the contributions made by different scientists towards nutrition. List out the elements that are required to carry out the process of photosynthesis. Describe the process of photosynthesis. Explain the synthesis of plant food other than carbohydrates. Describe the techniques of preserving the plant species. Explain the other mode of nutrition in plants. Know the plants that grow on dead and decaying matter. Explain the process of a special mode of obtaining nutrition. Know the colonies of algae and fungi. Interpret how nutrients are replenished in the soil.
Introduction. Before entering into the chapter, follow the instructions shown on the screen. Click each tab to know more.
In lower classes, we learned that all living beings need food. Food provides us energy for doing different activities and help us to grow and healing. We know that many fruits and vegetables are produced from a single plant. The quantity of production differs from plant to plant. Human beings, animals, and some other organisms depend on the plants for their food. Most of the plants require specific environmental conditions to grow. Do you know how a single plant is capable of producing many fruits or vegetables? The process of photosynthesis in plants. What are the materials required for the process of photosynthesis? How water flows from root to stem in plants. How exchange of gases takes place in stomata. The importance of sunlight to plants. What are saprophytes? What is the meaning of symbiotic relationship? Let us try to understand the process of production, factors contributing and regulating the growth and production in plants. Let us know about nutrition. Nutrition is the branch of science that deals with the nutrients. It is the process of providing or obtaining the food necessary for health and growth. Plants use inorganic minerals for nutrition. Roots absorb mineral nutrients as ions in the soil and water. Now, let us study about different modes of nutrition. Generally, organisms differ each other in their modes of deriving nutrients from the environment. Based on the mode of obtaining food, organisms are classified into two types. They are autotrophs, heterotrophs. Hence, there are mainly two modes of nutrition. They are autotrophic nutrition, heterotrophic nutrition. Now, let us study about the discovery of nutrition. Until 350 years ago, people believed the words of Aristotle, which he said 2000 years ago. He believed that plants are capable of producing everything from what they take from the soil. After him, different scientists have contributed their discoveries towards nutrition. Contributions of scientists towards discovery of nutrition. Jan Baptist von Helmont, Stephen Hales, Joseph Priestley, Engine House. Click each tab to know more. Jan Baptist von Helmont. Jan Baptist von Helmont is a Belgian scientist. In the year 1648, he conducted an experiment for 5 years. He took a small willow tree and planted it in a large pot of soil. Before planting the tree, he measured the mass of the dry soil and mass of the plant. Later, he covered the soil with a lid to prevent anything falling on the surface because it adds the mass to the soil. The lid contained holes so that the air and water could reach the roots and help the plants to grow. He left the plant for 5 years by providing only rainwater. After 5 years, once again he measured the mass of the tree and mass of the dry soil. The observations of his experiment were tabulated on the screen. From the observations, he concluded that the substances that are needed for the growth of plant do not come only from the soil. The plant grows because of the water it acquires. Stephen Hales. Stephen Hales depicted that the leaves of a plant function as the organs of transpiration. Transpiration is the process by which moisture is carried through plants from roots to small pores on the underside of leaves, where it changes to vapor and is released into the atmosphere. In other words, transpiration is essentially evaporation of water from plant leaves. Hales stated that plants exchange gases with their surrounding air. He was the first person to point out the necessity of light in plant nutrition. Joseph Priestley. Joseph Priestley carried out the sequence of experiments. He demonstrated that the process of respiration is reversed in animals when compared to plants, that is, according to him, if animals were making the air impure, plants were making it pure. Engine House. Another scientist named Engine House repeated Priestley's experiments under different conditions and found that only the green parts of the plants, when exposed to sunlight, make the impure air pure. Now, let us learn about the process of photosynthesis, the process by which green plants can make their own food, glucose, from simple substances like carbon dioxide, water, sunlight, and minerals which are present in the surroundings, is called autotrophic mode of nutrition. Organisms using this mode of nutrition are called autotrophs. Photosynthesis is the process by which plants prepare their own food. Photosynthesis occurs in the leaves of a plant. As the photosynthesis takes place in leaves, all the raw materials must reach there. Water and minerals present in the soil are absorbed by the roots and are transported to the leaves through the stem, branches, and finally to leaves. Plants get water from the soil through their roots. They absorb carbon dioxide which is present in the air with the help of holes present in the leaves. These holes help in the process of exchanging of gases. These holes are called stomata. Thus, the process of continuous exchange of gases in the leaves takes place through the stomata. We have learned that plants absorb water through their roots and the exchange of gases takes place through the stomata of the leaves. But apart from stomata, there are some other parts like loose tissues and lenticels on the bark of the plant through which exchange of gases takes place. Every green plant contains a green pigment called chlorophyll, which develops complex organic molecules from a simple compound in the presence of sunlight. As the synthesis of food occurs in the presence of sunlight, it is called photosynthesis, where photo means light and synthesis means to combine. Thus, it is clear that chlorophyll, sunlight, carbon dioxide and water are essential to carry out the process of photosynthesis. During this process, oxygen is released and the carbohydrates get converted into starch. The presence of starch in leaves indicates the occurrence of photosynthesis. The equation for photosynthesis is given on screen.
Knowledge check.
Here, let us perform an activity to learn about the need of sunlight in the formation of starch. Click each tab to know more.
Take a plant having destarched leaves, that is, a plant which is placed in the dark for 3 days. Next, wrap the leaves of the plant with black paper on which a design is cut without removing the leaves from the plant. Now, fix the black paper on the leaf with the help of clips such that light should not enter the dark part. Now place this plant in sunlight for a few hours. Later, detach the leaf that is covered by black paper from the plant and test it for the presence of starch by pouring iodine solution on it. It is observed that only the portion of the leaf which is exposed to sunlight turned to blue-black and the remaining portion of the leaf which is under the black paper remained the same. From the activity, we learn that starch is formed only at the parts where sunlight falls. The parts of the leaves which turn into blue-black color by pouring iodine solution on it indicate the presence of starch in leaves. Thus, we can say that sunlight is necessary for the process of photosynthesis to form starch. Here, let us know about the presence of chlorophyll in leaves other than green color. Generally, we find leaves of plants with different colors other than green. Does these leaves also contain chlorophyll? The answer is yes. In these leaves, a large amount of red, brown and other pigments mask the green color. The process of photosynthesis also takes place in these leaves. Also, you might have observed some green-colored patches in ponds or in other stagnant water bodies. These green-colored patches are generally called algae. Do you know why they are green in color? The presence of chlorophyll gives them the green color. Algae can also prepare their own food through the process of photosynthesis. Let us study about the synthesis of plant food other than carbohydrates. We have learned that plants synthesize carbohydrates through the process of photosynthesis. But do you know what carbohydrates are made up of? Well, the carbohydrates are made up of carbon, hydrogen, and oxygen. Carbohydrates are used to synthesize other components of food like fats and proteins. But proteins are nitrogenous compounds which contain nitrogen. How do plants obtain this nitrogen? The bacterium that is present in the soil converts gaseous nitrogen into a usable form and release it into the soil. These usable forms are absorbed by the plants along with water. You might have also observed farmers spraying or adding fertilizers to the soil which are rich in nitrogen. In this manner, plants fulfill their requirements of nitrogen along with other constituents. Plants can thus synthesize components of food other than carbohydrates like proteins and fats. Now, let us learn how to preserve the plants and its parts. Traditional designery art is a way of preserving the parts of the total plant like leaves, flowers, etc. Suppose the plants are not available in a particular location, then the physiologists collect those plants where they are available and make them in preservative form. This process is generally referred to as herbarium. The word herbar refers to a collection of preserved plant species. These preserved plants are studied by the physiologists and are also helpful for future studies. So far, we have learned that plants produce starch. Now, let us learn about other products that are produced by the plants. In the previous chapter, we have studied that our food contains starch, fats, and proteins. Similarly, all these substances are also present in the plants. First, plants produce sugar. Later, this sugar is converted to starch and to other compounds. But this conversion of sugar to starch involves many nutrients. The important nutrients include nitrogen, potassium, and phosphorus. Other nutrients are also required, but in minute quantities. These nutrients are termed as micronutrients. Plants absorb these nutrients from the soil. Studying of these nutrient elements by conducting experiments is very difficult. Now, we will learn about the other modes of nutrition in plants. Cascuta is a thread-like plant which contains yellow structures twining around the stem, branches, and leaves of other trees. It does not have leaves and chlorophyll. It takes food from the plant on which it is climbing. Just like animals and humans, this type of plant species also depends on plants for food. This mode of nutrition is called heterotrophic nutrition. Thus, the plants that depend on other plants for food are called parasitic plants. These plants develop special roots called haustoria. Haustoria are root-like organs that penetrate towards the tissue of a host plant and absorb food materials from them, and sometimes they may even kill the host plant. Here, let us know the plants that grow on dead and decaying matter. We often find some umbrella-like structures growing on the ground after rains. These types of plants are called saprophytes. These saprophytes grow on dead or decaying matter. Similarly, you might have also observed some black and white spots on bread pieces, pickles, whenever they are kept in moist places. These black and white spots are also a type of plant that are developed due to the growth of fungi. These plants do not contain chlorophyll. Generally, they absorb the organic material from the decaying matter. Let us observe the fungi on bread mold by doing an activity.
Click each tab to know more.
To observe fungi on a bread mold. To perform this experiment, the following materials are used. They are spoiled bread, permanent slide, plucker, cover slip, and microscope. Take a slice of spoiled bread and a permanent slide. Now, place some rotten part of the spoiled bread on a permanent slide by using a plucker. Next, add a few drops of water on the slide and cover it with a cover slip. Now, place the slide under the microscope and observe it. Whitish thread-like growth with masses of black, grey and green fine dotted structures are observed. The tiny thread-colored structures that are found on the bread are generally called mould. This mould develops on the spoiled material and it has the capability of producing new mould. The common bread mould plant consists of fine thread-like projections called hyphae and thin stems having knob-like structures called sporangia. Sporangium in singular. Each sporangium contains hundreds of minute pores. When the sporangium bursts, the tiny spores are dispersed into the air. Let us learn about the special mode of obtaining nutrition. The plants that can prepare their own food and also obtain their nutrition from the insects are called insectivorous plants. These plants are also called carnivorous plants. The leaves of these plants are specially adapted to catch insects. As these plants are green in color, they can manufacture their own food. These plants generally grow in the places where there is nitrogen deficiency. They meet their nitrogen requirements from insects. Some of the examples of these plants include Drosera's, Utricularia, Venus flytrap, and Nepenthes cassiana. In some plants like dal family, a type of bacteria grows on their root nodules. This bacteria acquires shelter in the roots of the plants and fixes nitrogen for the plants. This process of association is helpful to both bacteria and plants, and it is called symbiosis. The relation between algae and fungi starts when a type of fungus attacks the algal colony. This is an example of balanced parasitism. The algae and fungi live together in a symbiotic relation, forming colonies. These colonies are called lichens. The relation between algae and fungi starts when a type of fungus attacks the algal colony. This is an example of balanced parasitism. During the course of time, the algae survive and the fungus colony protects it from intense sunlight and conditions of drying up. The algae serve as the food provider for the fungus. In simple words, we can say that fungus gets food from algae and in return, it protects the algae from the intense sunlight and conditions of drying up. Let us study how nutrients are replenished in the soil. In some plants like dal family, a type of bacteria called Rhizobium grows on their root nodules. This bacterium acquires shelter in the roots of the plants and absorbs nitrogen from the atmosphere. It further converts nitrogen into ammonia and nitrate and helps the plant in their growth. Rhizobium cannot prepare its own food. It depends on plants for food and shelter. This type of relationship between bacteria present in roots and plants is called as symbiotic relationship. This association is of great importance to the farmers because they do not need to add nitrogen fertilizer to the soil. Types of leguminous plants include pulses like gram, peas, moong, beans, etc.
Follow-up work. Take up the following activities.
Get a sample of urea from the fertilizer shop or a farmer near your house or village. Sprinkle the urea near the plant whose leaves are turned to yellow color. Observe the changes in the leaves after sprinkling urea. Note all your observations in a notebook. Try to differentiate the plant before and after the sprinkling of urea. Collect a leaf. Take the peel from both sides of the leaf. Put the leaf under the microscope. Observe the number, size and shape of stomata. Note all your observations in a notebook. Attempt to draw the shapes of the stomata present in the leaf. Select any broad-leaved plotted plant. Take a cardboard and cut into desired pieces. Fix the cardboard pieces on the selected leaves. Put the plant under the sun for 7 days. Remove the cardboard and observe the changes in the leaves. Repeat the experiment on a number of leaves to get designer leaves. Note all the observations from the initial point of selecting the plant till you achieve the designer leaves. Visit a greenhouse if there is one near your place. Observe how they raise plants. Find out how they regulate the amount of light, water and carbon dioxide to grow the plants. Try growing a sweet potato just in water. Describe your experiment and observations. You have successfully completed the chapter Nutrition in Plants.
Topper's Classroom is always ready to providing free education to students worldwide. In every chapter we provide some free study material to the students which will be available in the description of video. Our mission is to empower each and every people of India to live their life in their own terms. And our vision is to make India healthy, wealthy as well as rich and to become a benchmark country worldwide.